Welcome to another episode from the Veterinary Research Channel. Today, we delve into the important topic of equine herpes virus type 1, a significant infection affecting horses worldwide. This virus, often referred to as EHV1, is highly prevalent and can lead to serious health issues in equids, most notably abortion in pregnant mares and a severe neurological condition known as equine herpesvirus myeloencephalopathy, or EHM. Given the ongoing impact of this virus on equine health and the equine industry, the American College of Veterinary Internal Medicine, or ACVIM, recognized the need to update their guidance on this critical disease. The previous consensus statement on EHV1 was published in 2009, and since then, continued scientific investigation has improved our understanding of this infection. The ACVIM Board of Regents tasked a panel of experts to revise the original statement, incorporating the latest research findings to provide the veterinary community with the most current information on the disease's mechanisms, diagnosis, and management. This updated consensus statement takes a rigorous, evidence-based approach, relying heavily on formal systematic review methodologies. To identify the most pressing challenges in managing EHV1 infection, the authors conducted a survey of large animal internal medicine diplomates of the ACVIM. The responses from this survey helped to shape 4 high-priority research questions that were addressed through systematic literature reviews. These questions focused on the effectiveness of vaccination, the efficacy of pharmacological treatments, the correlation between the level of virus in the bloodstream and the occurrence of abortion or EHM, and the best strategies for detecting EHV1 infection. Our discussion today will highlight the key findings from these comprehensive reviews and the resulting updated recommendations for managing this important equine disease. One of the primary areas of focus was understanding whether vaccination protects against EHV1 infection and the diseases it causes. The systematic review on this topic examined a large number of studies, ultimately including 35 that met specific criteria for evaluation. These studies investigated various types of vaccines, both commercially available and experimental. The effectiveness of these vaccines varied, depending on the specific type of vaccine and the outcome being measured. While some studies reported minimal or no benefit, overall, the evidence suggested that vaccination might reduce fever and signs of respiratory disease, and could also limit the amount and duration of virus shedding from a nose. This benefit appeared strongest with modified live vaccines and deletion mutant vaccines. However, the review found no evidence that vaccination completely prevents the presence of the virus in the bloodstream, known as viremia, and crucially, no evidence that it prevents the development of equine herpesvirus myeloencephalopathy after infection. There was limited evidence suggesting that killed vaccines might reduce the incidence of abortion. Despite these limitations, the consensus statement supports the vaccination of individual horses or groups of horses following a careful assessment of their risk of exposure and the potential consequences of infection. Vaccination is viewed as a tool that may help limit some disease signs and the spread of infection, but must be part of a broader biosecurity program to effectively prevent or limit EHV1. The authors emphasized the critical need for future research to develop safer and more effective vaccines. The second research question explored the effectiveness of pharmacological treatments in managing EHV1 infection. The systematic review on this topic identified 9 studies that met the inclusion criteria. These studies evaluated various interventions, including antiviral medications and immunomodulators. The overall findings indicated minimal to limited benefit from any of the studied interventions, whether used to prevent infection or as a treatment after exposure, in reducing the negative outcomes associated with EHV1. The consensus statement specifically notes that there is no evidence that pharmacological treatments given after the onset of clinical signs of EHV1 infection can prevent or alter the development or course of equine herpesvirus myeloencephalopathy. However, there was moderate evidence that the antiviral medication valacyclovir, when given to horses before EHV1 exposure, could limit the development of EHM, suggesting this strategy might be considered in an outbreak situation where early intervention is possible. The need for future well-designed studies to evaluate the efficacy of pharmaceutical interventions remains critical. The third research question investigated whether the severity of viremia, the presence of the virus in the bloodstream, correlates with the occurrence of abortion or equine herpesvirus myeloencephalopathy. The systematic review included 34 studies that examined this relationship. The findings generally supported the idea that viremia is typically present before either abortion or EHM occurs. This suggests that interventions aimed at eliminating viremia could potentially prevent these severe outcomes. However, the review could not establish a clear link between the magnitude or duration of viremia and the likelihood or severity of either neurological signs or abortion. The final research question addressed the best sampling strategies for detecting EHV-1 infection. This narrative review, which included elements of systematic review methodology, analyzed a number of studies evaluating different sampling techniques. When horses were sampled on multiple consecutive days, such as in experimental settings, both nasal secretions and blood samples showed similar sensitivity for detecting EHV-1 using a highly sensitive test called quantitative polymerase chain reaction, or qPCR. However, in situations where horses are sampled only once, which is more typical during disease outbreaks, nasal secretion samples were more likely to detect EHV-1 in horses with fever and respiratory signs, as well as in horses suspected of having equine herpesvirus myeloencephalopathy. In contrast, during abortion outbreaks, blood samples were more successful in detecting the virus. Experimental studies showed that EHV-1 could be detected in both sample types for approximately 9 days after infection, with nasal shedding typically preceding the detection of virus in the blood. The consensus statement recommends testing both nasal secretions and blood when possible. However, when resources are limited, testing nasal secretions alone is generally advised, except in cases of abortion and MARES, where blood samples are preferred. Beyond these specific research questions, the updated consensus statement also revisited the key questions addressed in the original 2009 statement, incorporating new information published since then. Our understanding of how and why EHV-1 targets the pregnant uterus and the central nervous system, and why some horses develop neurological disease while others do not, has been refined. Following initial infection in the respiratory system, the virus establishes itself in local lymph nodes and then spreads via infected immune cells to the blood vessels of other organs, including the uterus and the central nervous system. While a specific genetic variation in the virus's DNA polymerase was previously linked to neurological disease, more recent evidence suggests this association is not absolute and may vary geographically. Other viral proteins are being investigated for their potential role in the development of neurological complications, but their exact contribution remains unclear. Regarding the epidemiology of EHV-1, the updated statement highlights that the virus's ability to remain dormant in horses and reactivate later is a critical factor in its spread. Primary infection typically occurs early in life, but the exact percentage of horses that develop this lifelong latent infection is still debated. Factors that trigger reactivation in the field are not well understood. While subclinical shedding of the virus is relatively infrequent, large outbreaks of equine herpesvirus myeloencephalopathy continue to be a concern. The known risk factors for developing respiratory, abortogenic, or neurologic disease caused by EHV-1, such as age, stress, and certain environmental conditions, remain largely unchanged. However, studies continue to identify specific factors, such as increasing age and possibly breed, that may influence the risk of developing equine herpesvirus myeloencephalopathy. Large gatherings of horses have also been associated with significant outbreaks. Advances in diagnostic testing continue, with quantitative polymerase chain reaction remaining the preferred method for detecting EHV-1 due to its high sensitivity and specificity. While tests that can differentiate between different strains of the virus based on genetic variations are commercially available, the interpretation of these results in predicting disease remains cautious. Rapid, stall-side testing platforms are being developed, offering advantages in acute case management, but proper validation against standard tests is essential. The recommendations for using commercially available vaccines to control EHV-1 infection and disease are consistent with the original statement. Vaccination may be effective in preventing the major consequences of infection if it can prevent the occurrence of viremia. Despite the limitations in vaccine efficacy highlighted in the recent systematic reviews, vaccination remains a recommended part of a comprehensive biosecurity program, particularly for at-risk horses with a clear understanding of its protective limitations. The fundamental principles for controlling EHV-1 remain focused on preventing or reducing the likelihood of outbreaks and limiting the spread of disease when an outbreak occurs. Similarly, the key aspects of outbreak response, including early diagnosis, preventing further spread through isolation and enhanced biosecurity, and managing clinical cases, are unchanged. Recent experiences from major outbreaks have emphasized that infected horses can shed infectious amounts of the virus for a prolonged period, making strict isolation and biosecurity measures crucial. While the virus can persist in the environment for some time, proper disinfection is an important component of outbreak control. Finally, the updated consensus statement reiterates the limited effectiveness of pharmacological treatments for EHV, one infection beyond supportive care, aligning with the findings of the recent systematic review. In conclusion, equine herpes virus type 1 remains a significant challenge in equine health, and while progress has been made in understanding the virus, our ability to effectively vaccinate against it or treat its serious consequences remains limited. The authors of this updated consensus statement encourage researchers to consider the findings of their reviews when designing future studies, emphasizing the need for robust, well-powered studies with thorough reporting of methods and results to better evaluate the effectiveness of both vaccination and therapeutic interventions. Increased sampling over time and the sharing of individual animal data will also be crucial for future research and analyses. Thank you for listening to the Veterinary Research Channel. A link for the referenced paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.